slides over here. And the others, after Bernard's course, I'm sure you're able to generalize and know how you're going to have an exercise.
Excuse me, could you please write it again with bigger letters?
Okay, while Fernando's doing that, I need to calibrate a bit. So I'd like you to all look confused. Can you all try and do your best confused look? Pretty good. Now look as if you're understanding. See, that looked pretty much the same. It's going to be very... I was going to try and calibrate my speed based on that, but apparently that's not going to work. Okay. I'm talking about dimensionality reduction, which is something that interested me, I guess, when I started. I should say it's very true what Bernard says about meeting people places. I think I met Bernard, or saw Bernard for the first time in 1997 at the Machine Learning and Generalization School. So we've known each other, I guess, for quite a long time. And the same thing can, of course, happen at these summer schools. You know, you can build relationships with people you meet, and 15 years later, organize your own summer schools and have people attend. Okay, so I'm going to talk about dimensionality reduction, which is something I became interested in as a PhD student. I didn't do my thesis on it, but it struck me as something that was interesting and important. And shortly after that, an amount of work happened in spectral methods in machine learning, which I'm going to try and review. Part of what I say is tutorial, but part of it is polemic. I'm trying to push a certain point of view. You're entitled to disagree with that point of view. Not everything I say is, has to be considered right. I think it's right, that's why I say it. But it's also to encourage thought in yourselves. Other people take different perspectives from the one I present. What I wanted to first of all do is start with something I think I struggled with, which is actually what it means to think in high dimensions. When people tell you these things about how high dimensional spaces are, what that means in practice and why perhaps your intuitions aren't quite right. And then I want in this section to talk about linear dimensionality reduction and probabilistic linear dimensionality reduction. I'll also move on to the sort of non-probabilistic perspective in terms of distant matching given time in this session. Otherwise, I'll push that into the next session. I haven't calibrated the speed of this, so depending on how fast I'll go, different material will appear in different places. So just a bit of notation. So I'm following the statisticians in, when I talk about data, it'll be of dimensionality little p, which I think is confusing because it's a probability distribution, but that's what statisticians, oh, if ever I kick that, that's, the thing goes off, that's great. That's causality inference there, by the way. That's the second time I did that. But that's advanced stuff, we're not talking about that here. Latent dimensionality, I'm going to use Q. N will be the number of data points, and my data matrix will always be Y. So whenever you see Y, that's a design matrix containing data, so it's got N rows and P columns. So that's the definition of a design matrix. Again, it's statistics inspired. And I'm going to use the reduced dimensional representation, which I call the latent variables, but it's the low dimensional representation will be X. These guys will come in later. They're more for future slides. So just a little bit of notation. And then I'm going to use this MATLAB style notation to try and differentiate between columns from A and rows of A. So AI colon is it's the row of A, but as a column vector. And this is a column of A as a column vector. And then the element of A is Aij. Okay, so the source code and stuff is online. So why high dimensional data? It really dominates in machine learning, partially for some of the reasons Bernard was talking about earlier. I mean, not just the number of data points. As you increase that, it becomes hard for a human to look at a large number of data points. It also becomes hard to look at high dimensionality. So examples of high-dimensional data could be a customer in a database, and the features might include their purchase history, where they live, their sex, their age, their film preference, what tests have been done on them from a medical perspective. If you start trying to think of characterizing an individual in numbers, you're going to have a very large set of numbers. So a digitized photograph or video, it's got the pixel intensities, time, date, location of the photograph. One thing I like to play with is motion capture data. I like playing with motion capture data because it's very high dimensional, but if you have a good model of it, humans can recognize it. So it's something you're quite trained to perceive. If you see a model of a stick man moving, you know whether the motion's natural or not. Now that's 100 and something dimensional time series very often, but you can look at it and say, yeah, that's correct. So you're capable of dealing with high-dimensional data when it's presented in the correct form. That's something that shows that. So motion capture data is something that I guess I started working with, and I've created a MATLAB mocap toolbox. So if you wanted to use it yourself, you can just move it into MATLAB, and Jeff Hinton, the student, did that as well. So he demonstrates with mocap data, because it shows you when you've got a good model of high-dimensional data, it's very clear to a human when the human's moving naturally. So human speech, when I started in machine learning 15 years ago or more, there was a big overlap between machine learning and speech. So nowadays, that seems to be smaller, but I think it's coming back. In fact, people are doing sort of deep learning in speech recognition. It's a very interesting application domain, and there's a lot of opportunity, I think. Well, it's difficult because they do it very well, but I think it's an interesting set of data, but it's also high dimensional, and it's a time series. So web pages or other documents, so features could consist of frequencies of given words, that's bag of words models. Other features could be how those documents are linked. So I have colleagues who are in the language group in Sheffield that are analyzing Twitter feeds, and so each tweet, even though it's only 5 or 6 words, it's potentially from a language of maybe 16,000 words, and it's associated with information about where the tweet was given, who's following that Twitterer. Is that the right word, Twitterer? There's young people in the audience, you should know. And all this additional information. So that's high-dimensional data as well. Another area I work is computational biology, gene expression. So you can characterize the state of a cell by effectively the concentration and location of all the chemical compounds that are in that cell. If you think of the size of that state space, you have, I don't know, my memory is something like 30,000 genes in your body. Each of those genes has products, maybe in the form of proteins, many, many more proteins than there are genes. So if we think of the state of an individual one of your cells, you need, millions of variables to characterize its state at any given time, plus position, velocity of each thing. Very, very, very high dimensional. Okay. So how do we deal with complex data? So I've tried to simulate some complex data here, and this is very much starting from what goes on in a low dimensional, because typically people don't show you images of high dimensional data because you can't visualize it. So when I first started doing machine learning, if you looked at data like this, it looks complicated, but people had techniques for dealing with this. They had mixtures of Gaussians models. And here, with a circle locating the center and a little ellipsoid denoting the sort of covariance of the model, you've got a mixture of Gaussians fit to that data. Which you must think, and that's really good, because that looks quite complex in terms of data, and density modeling is something we want to do, understand the density of the data. And I tell you, that fit is actually perfect, because it's the model I generated the data from. So this isn't even a fit from the data, this is the true model. So once you've seen that, you sort of think, complex structure isn't a problem for mixtures of Gaussians, yeah? I mean, it can model these things quite well. People do a lot of effort into selecting the number of components, into dealing with the fact that there's multiple local minima, but fundamentally, surely that's the problem solved. Well, that's not the case, and it's not the case, I'd argue, because of the way you look at data in 2 dimensions is somehow sort of wrong. And a Gaussian may be a nice model in 2 dimensions, but a straightforward Gaussian, a Gaussian where you're not thinking about what you're doing, so if you just apply a Gaussian and fit its covariance in high dimensions, it's going to be quite a bad model very often. I still argue that Gaussians can be very useful, and you can do a range of things with Gaussians, but you have to do things like structure the covariance matrix in order to fit them. So why is it? You know, this is the sort of thing that I came into machine learning from a fairly naive perspective that in those days machine learning was about how the brain worked, and neural networks was how the brain worked. And then these bunch of people came along with support vector machines and messed it all up. Finally, with deep learning, they're going back to doing what we were doing 15 years ago, back to these sort of drawing these circles and putting lines between them. And I came in from a mechanical engineering background with this naive view that neural networks was where it's at. And then when I saw these statistical models of mixed Gaussians, I thought, well, surely they're very powerful too. But I think the reason that they aren't as widely used as they should be is because in high-dimensional data, they typically don't work very well. So these two-dimensional plots of Gaussians can be misleading, and our low-dimensional intuitions can fail dramatically. And there's a couple of major issues. That in high dimensions, in a Gaussian density, all that data actually moves to a shell. Now, I'm going to try and show you, visualize what I mean by moving to a shell. You may have heard this before. There is no data near the mean of the Gaussian. So one way of thinking about a clustering model like this is what you're saying about your data, and I like this way of thinking about it, is that each data point is really a prototype, that's the mean of the Gaussian, that has then been corrupted in some way, according to the covariance of the Gaussian. So you're saying you represent your data by prototypes that are the mean, plus corruption, which is the covariance, how they move away from the mean. But what this high-dimensional data stuff is saying, that in high dimensions, the prototype is never going to be observed, because you'll always be corrupted in a shell, some distance away from the prototype, which is kind of an odd thing. So how does that go on? So I used to hear this, and I used to think, okay, yeah, fine. And people would prove things about it, but I never had the intuition. So this is an exercise in trying to understand the intuition. And it's based around a Gaussian egg. Okay, there's another exercise. These are cursor dimensionality exercises in the old Bishop book. And what I want you to see in this egg, this is a one-dimensional egg. And then I'm showing a density across this egg. The yellow bit in the middle is the yolk. And then the white bit on the outside is the sort of albumin. And then I put a green bit right at the interface between the yolk and the white of the egg. So that green bit is, if you overboil your egg, like some places do, you see on the outside of it, there's a green covering. It's apparently iron sulfide, and it's why eggs smell bad, because they've got sulfur in them. So that green bit there, this is an overboiled egg. But it's got a relatively thin layer that makes an egg taste bad, actually. So you shouldn't overboil your eggs. But here, it's been overboiled, and the way we've set it up is that the iron sulfide is between 0.95 and 1.05 standard deviations from the mean, yeah? So between 95% and 105% of a standard deviation from the mean. So the yolk's up to 0.95 of the standard deviation, and the white goes from 1.05 to infinity. This is a big egg. I mean, well, a real egg would be a square density, right? And so here we're sort of fitting a Gaussian density across it. So that's our one-dimensional egg. So if we look at the one-dimensional egg, we find it's really nice because the yolk is my favorite bit of the egg. That's nice and runny and tasty. And 65.8% of your egg is the tasty bit. Okay, the white is sort of more healthy. It's got protein in it. 29% of your egg is, so near, close to 30% of your egg is protein. And 5% of the egg is this nasty iron sulfide bit that you don't like. Okay, so what happens in 2 dimensions, you can compute these volumes again. The volume of so obviously a probability distribution, so it's the total volume is 100%, and in 2 dimensions, you get 60% of your egg. So you've lost 5% of your yolk, yeah? And 7.4% of your egg is now the iron sulfide, and 33%, a third of the egg is now in the white. Okay, the reason, right, obviously because there's higher density here and lower density here, so even though you're sort of seeing, this is now the view above of the egg, and this is a Gaussian egg, so this is higher peak. There's a sort of mountain of egg there. So there's more density in the middle than there is on the outside. So, okay, but now if we go to 3 dimensions, the density on the outside goes up a bit. There's 10% of the density is now in this iron sulfide, 56%. So we've lost some of the yolk. But how does this behave as we go higher and higher dimension? Well, the nice thing, so we've seen a sort of odd effect, that seems to be going up. White goes up, yeah? Yolk goes down, and iron sulfide goes up. So does that continue to be the case? Well, what it turns out is we can actually compute the density of where the probability mass is. So if we're thinking of this egg in high dimensions, all I'm doing is I'm thinking of a Gaussian distribution where I'm sampling independently across axes with a fixed variance. So if the variance of the Gaussian is sigma squared, what it turns out is the distance from the mean, which is obviously from Pythagoras theorem, the square root of yi1 plus yi2 squared. So here's sample 1, here's sample 2. That's where we end up. That's our distance from the mean, yeah? So the distance is given by the square root. Now, I can't do much with the square root. Well, you can, but it's actually easier to work with the square of the distance. And that's going to be an ongoing theme in everything I do. So it turns out that the square of this distance is chi-squared distributed. Yeah, so that's a statistical known thing, used quite a lot. So each of these samples, their length is chi-squared distributed. So if you want the square of the distance of the whole thing, it's just the square of that, then it's the sum of some chi-square distributed variables. So that's rather nice because it's analytic. So I like to think of the chi-square distribution as a gamma because that way I don't have to remember lots of different distributions. But it's a gamma distribution in the way I parameterize a gamma, which is this form here. This is this density. That's the normalizer, and the important part are x to the a minus 1 e to the minus bx. That's the form of the distribution in x. In this form, the mean of this distribution is a over b. Sometimes people set it so that the b is underneath x and the main is a times b, but this is the way I like to do it. If you parameterize this distribution with a set to half and b set to 1 over 2 sigma squared, you get this scaled variant of the chi-squared. This writing of the chi-squared is showing that it's scaled by sigma squared. Yeah, so this is actually a scale parameter, and so the sigma squared appears underneath here. Yeah, so you've got a gamma distribution. The square of this value here is distributed as a gamma with a parameter, the shape parameter, set to half, and what I think of as the rate parameter set to 1 over 2 sigma squared. So the nice thing about that is what you're interested in is the distance from the actual origin of a sample after you increase the dimensions, yeah? And what it turns out is the gamma, rather nicely, if we want to add 2 gamma variables here and ask what's inside this square root here, that's also gamma distributed. As long as the scales of these 2 gammas are the same, which they are if we're assuming the same variance, it's gamma distributed with a shape parameter which is the sum of the shape parameters of these guys. So in this case it's 1, and a rate parameter which is the same as before. That's if these guys are independent, which I'm assuming they are. So that's important. We're assuming we're sampling each of our features of our data in this Gaussian egg is being independently sampled from a Gaussian distribution which is spherical. Yeah? So we can compute this cumulative density function, and indeed we can do it for p-dimensional data. So p-dimensional data, the sum over these yi squared, these yik squared, so this is the ith data point, and each dimension of it is distributed as a gamma with the shape parameter p over 2. Now that means that the expected value under this distribution is p times sigma squared. So what we'll typically do is we'll ask about 1 over p times the expected value, so that we keep the expected value of the squared distance, this is in p dimensions now, from the origin, to be sigma squared. So that means that in the case of a standard normal, it's going to be 1, yeah? So we'll be working with standard normals. So the scaling of that scales the rate parameter, and we've got now a density formula that says what will the distance, the squared distance, how is the squared distance from the mean distributed? Now that's nice because in our Gaussian egg model, we can now ask the question, how does that squared distance from the mean vary with the number of dimensions? You see this density is a function of p, the mean distance from the mean distance from the origin, which is the mean in this case. That's the entire volume of the egg. And you started out with a yolk which was 65% of the egg, and a white which was, I think, 28%. Now, initially, the volume of the white portion goes up, but then actually it starts to drop down. And you see the yolk drops down as well, until when you get to 1,000 dimensions, with what I've defined to be the aluminium, you basically have only points which are that distance from the mean. All your data sits in that shell. Does that make sense? So that's very counterintuitive versus what you think if you look at a Gaussian. Because you can see this Gaussian, the data's close to the mean, right? You know the Gaussian, the data's close to the mean. It's very obvious, right? Now there's a fun fact here that every marginal of a Gaussian is a Gaussian. So this is a two-dimensional joint Gaussian with a variance of 1, okay? But actually, no, it's not. It's a three-dimensional Gaussian. I'm just showing you the marginal of 2 points, two variables in that Gaussian, yeah? So it's a three-dimensional Gaussian. That means that when you look in these two dimensions, we're projecting the third dimension down onto the first two, yeah? So there's an amount of data that appears close to the mean here, but really in the third dimension it's sitting away from the mean, because it's a three-dimensional Gaussian. No, it's a four-dimensional Gaussian. So there are two dimensions, right, being projected onto the mean. So the bit you see at the mean is really two other dimensions where the data's away from the mean and it's being projected down to the mean. No. It was a five-dimensional Gaussian. Okay, I can go on like that for a while. And we can go on up to 1,000 dimensions. Let's say if it's 1,000 dimensions, this is a marginal distribution from a 1,000-dimensional Gaussian. 998 dimensions you're not visualizing, you're just seeing projected down to the mean, yeah? So that means that there's no data near the mean. If you go to, we could even, a Gaussian process is an infinite dimensional Gaussian. So you could even talk about an infinite dimensional Gaussian and look at these two parts of it, yeah? So then there's definitely no data near the mean, because we've projected infinite dimensions down onto the mean. So when you look at a Gaussian, the way I used to look at a Gaussian is, look, there is data near this mean. Whatever people say about what's happening in high-dimensional spaces, I can see for myself that there's data near the mean. But remember, you're looking at potentially a projection of a very, very high-dimensional Gaussian down onto those two dimensions, which means that all this data in that high-dimensional Gaussian must be nowhere near the mean. Yeah? So in fact, this approximation is so good that in many areas, they will use a Gaussian distribution, taking it to high dimensions, as an approximation for a sphere. Yeah? So in very, very high dimensions, if you want to have uniform data distributed over a sphere, you can approximate that with a Gaussian. And that's a sort of known physics trick. Works very well as dimensions go in the thousands. So Gaussians are bad distributions, apparently. So the other thing that happens is interpoint distances. So in one slide, this is sort of trying to show that the same thing happens with the distance between data points if they're sampled from this Gaussian density. For very similar reasons, it comes out very quickly if you think about it. So if you've got two data points both sampled from the same Gaussian, then subtracting one off the other leads to another Gaussian density, and squaring it then again leads to this gamma density. So that's just the only new step, is subtracting one from the other, and sigma squared becomes 2 sigma squared. Everything else stays the same, and you can see that these data points are gamma distributed. The interpoint distance is gamma distributed, with the parameters of the gamma being p over 2 and p over 4 sigma squared rather than 2 sigma squared as we saw before. So I like this representation because it's very easy and very common to compute inter-data point distances, yeah? To do k nearest neighbors or whatever else. And in fact, I'm going to base a lot of the material on computing distances like this. So the dimension normalized between points is drawn from a gamma, and the mean is 2 sigma squared. Now the interesting thing about this, the variance of this distribution is 8 sigma squared over p. So as p goes high, this variance goes to 0. Yeah? Certainly relative, if you look at the mean divided by the square root of the variance, you've got this scaling of p that as you go to very high dimensions, what you'll see is that all the interpoint distances of your data set are the same distance apart. yeah, in this Gaussian egg. So any sample you take will be equidistant from every other sample, which is an annoying characteristic if you're trying to do things like k nearest neighbors, because as you change k, you'll find that if you look at the volume of neighbors you're considering, as you increase the nearness of the neighbor, you suddenly go from no neighbors to all your points are neighbors. This basically says that for this density, every data point is a neighbor of each other in high dimensions. Okay, so now that seems to be a bad thing about Gaussians, so you shouldn't use Gaussians, you might think. But actually, for any data where you've got independence over features, the central limit theorem applies to these sums we're taking, and the same results come out. They just don't come out quite so nicely and analytically in the low sample area. You get exactly the same thing going on. The variance about the mean scales as P1 over P. So any distribution you sample from independently will lead to these different effects. If you want to know how it differs, well, the Gaussian in this sphere, it's like if you, one of the reasons I believe we're here is because there's a large telescope here, and Bernard likes large telescopes. But it's useful for analogies as well. If you're standing on the Earth looking out at this hypersphere, okay, so it's high dimensional space, of these Gaussian samples, how will it look? Well, all the samples will be uniformly distributed across space, a bit like the stars. They're not quite uniformly distributed, yeah? But if you're standing at the center of this hypersphere, so it's a three-dimensional sphere here, but it's a hypersphere in general, looking out at these data points, they'll look like stars distributed over space. It'll be beautiful. If you use non-Gaussian densities, sub-Gaussian or super-Gaussian, in the hypersphere, the points will cluster at either alongside the axes or at 45 degrees to the axes, depending on whether you've used super or sub-Gaussians. So they just won't be uniformly distributed over this hypersphere anymore. It won't be quite as pretty. But the same problems will exist. So these distributions behave very counterintuitively in high dimensions. We can compute these densities of square distances analytically for the Gaussian case, and for non-Gaussian independent systems we can invoke the central limit theorem. So let's see how this applies for some real data. What we're going to do now is we'll see that there's this problem, and we can compute the theoretical interpoint distance between two data, and it's valid because of the central limit theorem for any data set if the features are independent. So here's some example data sets. Well, let's just test our intuition. So this is samples from a 1,000-dimensional Gaussian, and what I'm showing is the distribution of square distances between each data point. Yeah? And what I'm showing you then is a histogram, and then bang directly over it is the fit of that gamma distribution we derived before. And look how nicely the theory fits to data. So that's good. We can sample from a Gaussian, create a data set, 1,000 samples, so that's 1,000 data points, so around 1 million interpoint distances in this histogram. Okay, and now I've used fewer samples here, and the fit's still good. So this is 10,000 interpoint distances, the fit's still excellent. Okay, so let's take that, and let's apply it to real data, so that we can get our NIPS paper. So we have to have a little artificial example, then a real data example. Okay, the real data here is a little bit artificial as well, but this is a famous data set, which is so-called oil data. It's some oil flowing in a pipeline, and it's from Bishop and James, this 93 paper. It's widely used as a benchmark in dimensionality reduction, and it's oil flowing from a well, so it's got oil, gas, and water in it, and they, according to the speed, I guess Reynolds number and stuff like that, they'll mix in different ways. In turbulent flow, you basically get this homogeneous, everything mixed together. But then there's two types of laminar flow that occur. One is gas, oil, water, and then the other one I think is gas, oil, water. I hope I've got that the right way around. It might be gas, oil. No, it must be gas, who knows. Anyway, it's like that, sort of. Now, you've got 12 measurements, which are gamma ray densitometry measurements. So these are measuring the density across here. And this is a classic sort of learning problem, because although clearly if you see the density on each of these, you should be able to determine which domain you're in, the physics of the thing is a little bit complicated. So in fact, turns out this data is simulated if you read carefully the original paper, and it's simulated by understanding the physics of these systems and what these things would read. So a little bit of a warning there. it's simulated data, but it's from a realistic physical system. So what you can see here is you've got, I think, 12 sensors in total, measuring the density and the cross-section of these pipes. So that's 12 readings. So if we now compare the truth to what we theoretically expect to happen, we see that this is the theoretical curve for the inter-data point distances, and this is the actual histograms. Okay, so that's not too bad. There's a little bit of a mismatch. P is 12. The predicted variance of these interpoint distances is 0.66, and the actual variance we've observed is closer to 2. So it's quite a lot larger than we'd expect, the variance of interpoint distances. So anyway, okay, let's not worry too much about that, and let's look at another data set. I told you I like motion capture data. This is motion capture data where the data is only 55 frames, and there's 102 data points, because there's 34 points on the body, so they put balls on the body at 34 places, and there's an X, Y, Z location for each of these balls, yeah? So that's 3 times 34, 102 dimensional data, 55 data points, and it's from Ohio State University. I use this little data set a lot. It's a nice small data set, and one thing I like about it is the guy changes angle as he runs. So there's two things going on. There's one is periodic because he runs. It takes about 3 paces, and the other one is some sort of change in his angle, which is sort of overlaid on top of the periodicity. So let's do the distances for this data and the theoretical curve. Okay, P is now much larger, so the expected variance around the mean distance, which is 2, I've normalized all this data so each dimension is variance 1, so the expected square distance is 2. But what we observe here is that the variance is 1 versus the predicted of 0.07. Microarray data, so this is 6,000 genes and 77 experiments. It's a classic early microarray cell cycle data set. It's measurements in yeast of these 6,000 genes across different time series with different things being done to the yeast. So the 77 experiments are done, each in different time series, and 6,000 genes. And now if we look at the theory, the effect is even worse. Look at this guy up here. His predicted variance is less than 1%, 0.00129, but the actual variance of the sum of the squared distances is 0.694. So that lesson goes on. Here's a vowel data set. We see the same thing, less pronounced than the gene expression. So very high dimensional data is basically not reflecting what we saw in the theory. The situation for real data does not reflect what we expect. So what's going on? Why does real data not conform to maths? It's very naughty. Okay, something's gone wrong in our assumptions about what's going on in the maths. So let's look at P being 1,000 again, 1,000-dimensional data set, artificial, but we get a similar effect. This is a Gaussian with P being 1,000. But I've given this Gaussian a very specific low rank covariance, where W is 1,000 by 2, and then I've corrupted it by a little bit of noise. Okay, sorry, that's a bit low. So the form here is the covariance of this Gaussian is WW transpose plus sigma squared I, where W is a matrix of 1,000 long and 2 columns. So it's basically, this is rank 2, the covariance, plus some diagonal term to make it full rank. Now, I've taken the noise to be 0.01 and sampled this matrix W from a standard normal. Now, interestingly, if I now apply the theoretical curve, assuming a dimensionality of 2 for the data, the fit is bang on. What's going on? All our assumptions in that material I gave you at the beginning was that the features are independent. If you have 1,000 independent features about something, let's consider the case where they're binary. 1,000 independent binary facts about someone, that covers 2 to the 1,000 people. The space that it's covering is absolutely enormous. If you have that degree of specification on the data, something really weird is happening. In practice, real features are massively correlated. Yeah? So all this cursor dimensionality stuff is actually rubbish. It's rubbish from the fact of when you're thinking about the data. If it was true in your data, there would be no information in your data. It's what we call noise. The only thing we assume full independence for, across all data points and across all features, is noise. What we do in practice is we assume underlying regularities in our data, particularly, for features and everything else. If we didn't, we would not be able to learn from the data. The worst case predictions from learning theory would apply. But in practice they don't, because real data is correlated like this. This is a simple correlation, and we're going to talk about this correlation first, how you fit models that assume this correlation. There's not an invalid lesson though. It doesn't mean that you don't have to worry about the curse of dimensionality, because it's actually associated with your model, not your data. Yeah? We were sampling from a model that had this independence characteristic. If you fit such a model that has this independence characteristic, your model is covering a vast space of data. It's going to be difficult to extract information from the data. So there's something wrong with models like that. But I think it's endemic in machine learning and statistics to claim that this is a characteristic of the space. It's a characteristic of the model plus the space. Yeah? It's the worst case thing that could happen in the space. But then if someone brought you that data, which has that nasty characteristic, you really want to be asking them, what rubbish have you just given me? There's no information in this data. Because there really isn't. That's what these lessons are about. If there's information, if there's regularity in the data that you can extract, those things won't be true. Okay, so one thing I hope to do is put some of the curse of dimensionality, I just think it's, in a large sense, it's a myth. A lot of the things people extract from it, Bernard touched on it earlier, saying you shouldn't expand things into high dimensions. That's what the statisticians say, because it's a bad idea, right? Making your feature size infinity, that's always going to lead to problems. No, it's not, because of the structure of the model, because of the capacity control. That's the way Bernard was talking about it, yeah? I'm saying the same thing. It's about your model, yeah? Not about your data set size. It's a very bizarre thing, if you think about it, for someone to come to you, you're going to fit a model, and someone's bought you, here's your client, he's walked into your room and he says, there's the data set, I've found out 100 things about these 50 people, please can you fit your model? And you think, okay, that's difficult because P is large and N is smallish, but I'll try and fit a model. And then the guy goes, oh, I've just heard, I can tell you 100 more things about those people. Would you like to know? And then you say, no, don't tell me, because then I'll have an even larger P. I don't want to know anymore, because it will mess with me. The data's worse. How can that be true? Because it's not true. Those 100 things will not be independent of the previous 100 things. You'll get a richer model of the person, so on, so on, so on. Okay, there'll be more stuff about that later on. Okay, so where does this low rank covariance come from? It comes from a low-dimensional approximation for the data set. That's the connection with dimensionality reduction. A simple low-dimensional approximation, a linear low-dimensional approximation. If we made this CWW transpose plus D, I made D sigma squared I before, then that would be known as factor analysis. The form I used is known as probabilistic PCA. Okay? Probabilistic principal component analysis. And it's a linear mapping from a Q-dimensional latent space. Because I'm sort of build probabilistic models, I think of the low-dimensional thing as latent variables, unobserved things, to a p-dimensional data space. And then we corrupt that mapping, so we do a linear mapping from that, and then we corrupt it by independent Gaussian noise. That noise is high-dimensional. That is something we can't, that's almost like, it's one way of thinking about noise. It's the nasty, difficult stuff that we can't extract information from. The earliest people who defined noise were people like Laplace and Gauss, who were looking at planetary motions through the atmosphere and imperfections in their telescopes, and they said, these are things I can't account for. Let's assume they're independent random things being added to each observation, and they're unstructured. There's something that they can't deal with in the structure. Structured noise is very interesting, but it's not something we're assuming here. And then what we do is we marginalize these latent variables using a Gaussian prior. That's how we do probabilistic PCA. So just a reminder on the notation. So Q-dimensional latent space, P-dimensional data space, n the number of data points. Y is our data in this n by P matrix. That's important because it turns out if I write Y transpose, that's an inner product matrix. Centered data is like our data with the mean subtracted off. So that's something we'll use a lot. And then the latent variables are X. And then we're going to use this mapping matrix which maps from a P dimension to a Q dimensional data space. So if, in effect, when I go from, okay, so just about the notation, why am I making a fuss of this notation? Because when you see this form arising, the centered data set in this form, that's a covariance. Yeah? So I sometimes write that as S. So that is equal to that. And then if you see this inner product matrix, this YY transpose, that's an inner product matrix, which has this interpretation as a linear kernel sometimes. So those two are features of the way I've defined the data. And this is how we're going to model the data set. So we find the lower dimensional plane embedded in a higher dimensional space. The plane is described by this matrix W, which is in that space there. So we go from this low dimensional space, two-dimensional in this case, to a three-dimensional space by multiplying the vector x by W, which gives us a three-dimensional response, and adding a mean to it. If we also add noise, then we have this sort of form. So for each data point, we're assuming the relationship between the data and the latent variable is given by this equation. Now the way we write that probabilistically is as a Gaussian density if this is Gaussian distributed. So if this is Gaussian distributed with variance sigma squared, so this is that nasty high dimensional thing, it's independent across features, then what we get is the probability of y given x has a mean, well, independent across data points, we assume, and it has a mean given by wxi plus mu, and then it's corrupted by some noise of covariance sigma squared. Yeah? That's the likelihood. Now these x are things we don't know. So the parameters in our graph are w. I know there's a big tendency to use graphical models a lot in machine learning. I used to like it too, but now I don't know if it's something with age. I just look at them and I can't understand them because there's lots of circles and plates and things, and I find it hard to see what the conditional independencies are. But this is an easy one, okay? So y is dependent on x and w. It's also dependent on mu, but I didn't include it in the graph because I didn't want to confuse you. So the latent variable approach is that these x are nuisance variables, and we want to get rid of them, because we don't know what they are. We don't know what the underlying causes are. I mean, historically, principal component analysis was invented by who? Anyone? Hotelling. Good man. If someone said Pearson, they would have been wrong. Pearson invented the same algorithm, but for something different, and these two ideas have been conflated. I want to deflate them. I want to inflate them. I want to flate them. Hotelling was following the ideas in psychology at the time, which were known as factor analysis. Yeah? So Hotelling was trying to put on what he thought was a more sound mathematical basis factor analysis. And this is 1930s, I think 33 or 36. And his paper is really clear if you read it today. And he thinks of principal component analysis 100% in this way. As you've got some latent factors, which, you know, as Bernard's saying, you have these intelligence tests, what comes next in the series. Those are the sort of things they were scoring, and then they try and work out the latent causes that you're a psychopath and your IQ is 5. Yeah? So those are the X's, the underlying variables. In psychology, this was very important. And Hotelling came along and said, this is very nice, what you're doing, but here's a mathematical way of doing it. I don't want to call it factor analysis, because factor means something else in maths. So I'm going to call it principal component analysis, but it's inspired by the social sciences, and he thinks of these types of models. So principal component analysis is another type of factor analysis. And I'll sort of explain in a minute why I think it's a shame he changed the name, because, well, as you'll see from the maths. So we define a Gaussian prior over this latent space X, and this is what, oddly, although this is what Hotelling and did describe, he only looked at the noiseless case in effect. Now, it took till 1996 when the maximum likelihood solution for the noisy case was given by Tipping and Bishop, and Roweis also proposed a model around the same time, 96, 97, 98. Their papers come out at a different time, but it was all contemporaneous. So if you've got this prior distribution, what you do is you use integration to get rid of it. So we take the prior, we multiply it by the likelihood, and we integrate over x, and we get this marginal likelihood, which is the form of the Gaussian we use to sample from. So it's just a Gaussian with a mean of mu and a covariance which has that structure. Now remember what I said in the beginning. Be aware of Gaussians. But Gaussians with structure are good. In fact, that's all I do. Gaussians with structured covariances. Different structures on the covariance. And this structure is a low rank plus a diagonal. And now you need to optimize the likelihood of this. So the way we indicate that as a graph is we remove the marginalized variable, and there it is. Now, why do I think it's a shame he changed the name from principal component analysis to, from factor analysis to principal component analysis? Because it is a factorization of your data. It's factorization of your covariance. Yeah? WW transpose is a factorization. So even though that wasn't factor in the sense the psychologist meant, the actual underlying algorithm has this factorization of the covariance in it. Yeah? We'll see what factorization that is in a moment. I'm going to assume that you know that fitting the mean of a Gaussian is given by, well, the mean of the Gaussian is given by the sample mean. And so I'm going to do that. Look, that's me fitting the mean. Okay? So I'm centering my data. It's a common trick. You can do it if the noise is constant. You can't do it if the noise is varying or if the student T noise or something like that. For Gaussian noise, constant variance Gaussian noise, you can do that trick. So we'll apply that trick generally. So what Tipping and Bishop showed, the paper's 99, but the work was earlier, takes a while to publish in stats journals, is that the maximum likelihood solution is given by this form here. So if C is that covariance, then the log likelihood is given like this. So we rewrite the form of, this is a way you can rewrite the argument of the exponential in the Gaussian. You can turn it into this trace times the inverse covariance times y transpose y. And the reason for doing that is that that guy there is the covariance. And it turns out that the maximum likelihood solution is given by the eigenvalues of that covariance. So let's just go through a little bit on the details for that. So how do you solve this sort of thing? Well, you just take the gradient of the log likelihood and you do matrix derivatives. So the gradient of the log likelihood is given by this form, where the covariance is appearing, the sample covariance is appearing inside the covariance of the Gaussian. In a normal case, this W wouldn't be there. You would be taking the gradients with respect to C, and you would just recover that the covariance of the Gaussian is equal to the sample covariance. But here, because we've got this W constraining the covariance, it appears on this side of each of these C minus ones. So I don't know how that extra slide came in. If we pre-multiply that, what we're going to do is look for fixed points of that. So we set that to 0. And then we can pre-multiply it by 2c, which gives us that W is equal to this form, bringing that on the other side and moving the minus, and bringing the n by the covariance. So that is now the sample covariance, yeah, because it's 1 over n. Now this you still can't solve, and the trick that Mike Tipping noticed, and this is important, I want you to know that this trick is in there because it's what prevents factor analysis being solved in this way. There's a fixed point solution for principal component analysis, and there's no single one-step fixed point solution for factor analysis. It's an iterative algorithm, factor analysis, whereas PCA is a one-step. So you substitute W with its singular value decomposition. So I've written that in U, L, R transpose. So R is a rotation matrix, and it's going to turn out that you can't identify this rotation matrix for PCA. So that would imply, if we substitute that in the covariance, and this is why you can't identify R, is WW transpose is U, L, R transpose R, which is because it's a rotation matrix, a square rotation matrix, just becomes the identity. So you get U, L squared, U transpose. You can then use the matrix inversion lemma to show that this is the case. The inverse of the model covariance times W is equal to this form. That's being expressed in terms of re-expressing this W and the C minus 1 in terms of this eigenvalue decomposition. And once you've got that guy, that allows you to substitute into the original data and show that the solution, the maximum likelihood solution, is given by this sample covariance times U, E being equal to U times this sigma squared plus L squared, where L squared were those singular values from the singular value decomposition. Now that is recognized as an eigenvalue problem, yeah? And it's an eigenvalue problem on the covariance of the data. So I've moved through that quite quickly, but the key point here is you can't express this guy for factor analysis. You can't do this rewrite. You can try it at home. The reason is because this instead of being plus sigma squared I, it's plus d times I. So the effect that has, see if I can do this. I forgot to plan this bit, so if I mess it up. So if you've got W transpose plus sigma squared I versus W transpose plus D. So that's factor analysis. That's PCA. You'd think it was a lot more complicated than that if you read a lot of text on it, but that's basically it. That's diagonal. And you want to fit all these things. Well, when you do the single value decomposition, sorry, I had it as L, didn't I? UL squared, U transpose, plus sigma squared I. So the trick that's often used here is that this is also true. Yeah? So that works, but this doesn't. Sorry. So it's annoying, isn't it? I mean, how little simple things cause problems. You can't write this. So this is now sigma. Yeah, that's full covariance. This guy here is full covariance in order to write it in that way. Because you've rotated in one. Okay, another way of, what does this mean physically? What are eigenvalues? Eigenvalues are simply the axis aligned. So the Gaussian noise of the factor analysis is not axis aligned. Is axis aligned. And the Gaussian noise for PCA is spherical like that. So this is factor analysis. And here you've got 1 sigma I squared, sigma 1 squared, and you've got sigma 2 squared. Yeah, different in different dimensions. PCA is circular. What happens if you rotate a circle? Yeah, nice, isn't it? What happens if you rotate an oval? It develops into a full covariance Gaussian. Yeah? So the fact that that problem exists, this trick of being able to rotate a circle and it's still, you don't change the nature of the spherical noise if you rotate it. That's why you can do this nice, simple eigenvalue decomposition on the principal component analysis. You can't do it for factor analysis. Okay, good. So that leads to this eigenvalue decomposition. And actually, while we're at it, since I've got the... what do I interpret this as? Well, what's going on is you've got your covariance of your data. What you're trying to find is the principal directions. U is the directions that will allow you to axis align this data set, yeah? So this is like U1, and this is like U2. So that's what you're looking for. It's the covariance, the sample covariance from your data is given by this circle here. And then you do an eigen decomposition on that, which gives you the principal axes. Principal axis comes from principal component analysis of that covariance. Now, the rotational invariance is very interesting because the rotational invariance we had before, remember it was U, L, R, transpose R, L, U transpose. Yeah? Okay? So these things convert to W. I shouldn't have said that's U because it's actually W1 because it's multiplied by its eigenvalue to give it its length. U gives the direction like that, and the length is given by the eigenvalue. This rotation guy, what he does is things like this. All these are valid solutions for W. Yep. So these two, I should have used different colors there, shouldn't I? So let me try that. These two would be one set of solutions, and these two would be another set of solutions. They're rotated versions of the orthogonal set. So we tend to use the orthogonal set. They're known to be orthogonal because in the eigenvalue decomposition, that's a set of what I think of as orthonormal vectors, and that's a set of scales. Yeah? So these guys must be at right angles to each other. Good. So hopefully that's given some of the intuitions I have, or I think of myself, about principal component analysis. But just to finish what's going on in the solution, U are the eigenvectors of the covariance, and sigma squared plus L squared are the eigenvalues. Now, in traditional principal component analysis, you just need L squared. Sigma squared is taken to 0. That's the noise. And it's, in Hotelling's work, the noise was considered at 0. But in probabilistic principal component analysis, you add this sigma squared onto the eigenvalues. Now, what that's saying is that means that you're constantly adding to your data. You're smudging it. So if we were doing one-dimensional principal component analysis like this, okay, so we're looking for the first principal direction, then when you add the noise to it, what you actually do is you add a second component to it. A little bit of noise out in that direction, and you add a little bit on the end as well. Yeah? So the eigenvalues you get in principal component analysis, probabilistic principal component analysis, are slightly different from the ones you were getting if you get PCA, because you add this little bit of noise at the end. But you also add, it means it's a proper probabilistic setup. Because if you just were to take the vector WW transpose, the one-dimensional vector, that's not a valid covariance. It just defines, well, it's a semi-definite covariance. It defines a line. So adding the little bit of noise across the diagonal adds a little bit there and a little bit there. So you end up having to subtract that off in your solution for PCA. That comes out of the maths, but that's my intuition. The maths, I think you can see in the papers. I try to give you the intuition because that's what it took me longer to work out. So further manipulation shows that we can constrain this. Then the solution is given by the largest eigenvalues. What you don't see here is that the eigenvalues you need to retain, if you're reducing the dimension of U, are the largest ones. But that's, you can show that as well. So the Q largest eigenvalues. So some further work shows the principal eigenvectors. And then it turns out you can also work out, this requires quite a lot of further manipulation, that the maximum likelihood value for sigma squared is given by the average of the discarded eigenvalues. Okay, so if you want to know what sigma squared is, you have to take the average of the discarded eigenvalues, and that's sigma squared. So this is some intuition people have always had about PCA, that somehow they look for these elbows. People talked about elbows, finding dimensionality. These are an amazing thing that don't actually exist in real data. But the idea is if you do PCA, you look at the eigenvalues along this axis here, and you see some sort of elbow like that. So the eigenvalues go down, and then this is the elbow, the mythical elbow. And the idea is that this is the noise level, and then you retain any of those components. Okay, so PCA is making this more explicit. It's if you really took the eigendecomposition of that matrix form, WW transpose, plus sigma squared I, you really would see this. This would be sigma squared. Yeah, and there would be a constant eigenvalue, and this would be the dimensionality Q of these matrices here. Yeah, so it's encoded in this model that's being fitted to the data. But in fact, if you try and fit the number of components, it will always say that everything's a component. The maximum likelihood is found by including all components. There's no magical fit where it will find this noise for you. So you either have to do one of two things. One is the classic thing we all know and love, which is to select the latent dimensionality, Q, and then fit sigma squared. It's the average of the discarded eigenvalues. But the other cool thing you can do, which people often don't understand with PCA because they may not understand probabilistic PCA, is you can set sigma squared to some known value, let's call it A. And if you do that, then that will determine the dimensionality of the latent space for you automatically. In fact, what it turns out is, bear in mind this, so this is UQ, the first Q eigenvectors of the sample covariance. L is a Q by Q matrix. It's given by the first Q eigenvalues minus sigma squared I. If I set sigma squared to some value A, then this will be complex for any eigenvalue which is smaller than A. That doesn't mean there's a complex solution. What you can prove is that you should discard all dimensions who have a smaller eigenvalue than that of sigma. Yeah? So in that case, it's somehow saying that the physical analogy of that is that your data looks like this. It's only a covariance that looks like that. You're setting the variance, the noise variance, something like that, larger than that eigenvalue. And it will say, no, there's only one dimension in this data. It will switch off that dimension which is smaller in standard deviation than your noise variance. So that's something you can do, and we ourselves use that to effect in a model where we estimated the noise level by separate means on gene expression data. So we were doing PCA on gene expression data, and we had a way of estimating the noise in each gene expression measurement, and then that meant that the dimensionality of our PCA was automatically determined. So if you're sort of aware of that, that can be a useful trick. Okay. So that's principal component analysis, and those data sets I mentioned earlier, I'm just going to sort of quickly show you PCA on those different data sets, and then we'll do a 5-minute break. So this is that stick man walking. I told you he did 3 strides. He actually starts from a strange pose. He starts from this pose, and this is the start point here. And it's actually, if you look at the third principal component, you'll see that's disappearing off into the distance. So we're just looking at the first 2. The first 2 represent the strides. So these circles here, he's actually changing the angle of his run. That's why they don't lie on top of each other. The data's set so that he runs in place, yeah? So I removed the mean, so he just runs in place. Otherwise, it would just be a long stream of data. And what you can see is basically that because of the changing angle of run, the different strides don't quite align on top of each other. I'll show you this data set in the next session as well. I probably should have shown you this one, but I forgot to do that. So here's that oil data, and these are the model doesn't know the different flow regimes, but this is the homogeneous flow regime, the blue. And these are, one of these is annular, and one of these is layered, stratified, yeah. So they look very similar because actually the readings, the reflections that occur in the readings of the surfaces are kind of the same. Here you get lots of little reflections of little bubbles, so they tend to live in these different regimes. So you can't quite separate there's some blue under here, you don't quite separate with 2 principal components. This is data, this was the val data that I didn't talk much about. How I've separated these is into men and women. So red crosses are men, green circles are women. This is data I used at a tutorial at Interspeech. So I actually had, some of the names here are people who are known in their community because it's speech people. But I also labeled, there's these 3 accents here are all South Yorkshire accents. They're from our admin staff in Sheffield. And they sort of come close-ish together. Some of the undergraduates are close to them, others are some far away. So you see that there's some correlations in the structure and the data. This is me here. I'm worryingly close to the women. But that's because PCA is a limited algorithm. Really, my voice is very masculine. It just didn't pick that up. And John Barker, who I gave that tutorial Interspeech with, is over here. So we're apparently very close together. So this is based on the vowels we make. It's a hidden Markov model fit to our speech. And then I just took the vowels and the parameters of the hidden Markov models associated with the vowels and doing PCA on that. This is the gene expression data. I don't know if this should separate or not, but what I did is I labeled the different time series. I told you it's this yeast cell data, and it's some different time series conflated together. So each time series I've actually labeled with a different thing. So this is one of the time series, blue crosses. The purple squares are another time series. The red crosses, I think, aren't even time series. They're just one-off experiments. And the cyan asterisks are a separate time series. They're overlaying a bit, but blue seems to separate on 2 sides. And is the structure there? I don't know. But it's one way of visualizing it. Okay. So what's the point in the probabilistic approach? Why wouldn't we just project with regular PCA? So why do probability? This is the standard question again, and it's the standard answer that you always put in your papers. It's easy to extend to mixture models. So instead of doing mixtures of Gaussians, the first thing you should be doing is either mixtures of PCA or mixtures of factor analyzers. If you've got a lot of data and you do a lot of restarts, you'll find that mixtures of factor analyzers proposed by Zubin and Matt Beal some time ago is a very good, the Bayesian mixture factor analyzers that they use, which also learns dimensionality, is a good model. And Mike Tipping's was really good at fitting these mixtures of PCA, and he could get some really good density models. Because what you're doing there is you're lining up little linear manifolds, yeah? So each of these things, as you do a mixture model of this, if there's enough data, it fits these sort of small correlated areas. So what you're saying in those cases, and this is a much more reasonable model, in your high dimensional space, you've got a prototype, and the prototype is corrupted locally in a linear way, yeah, with some dimensionality. That's a very reasonable model. You may need a lot of data to fit all those local linear patches, and that's the weakness of it, but it's a very good way of modeling data. So that's lesson 1. If you're going to do mixtures of Gaussians in high dimensions, don't. Do mixtures of probabilistic PCA or Bayesian factor analysis. Excellent standard model for comparison against. You can do model selection with Bayesian treatment of the parameters, so learning the dimensionality. If there's time in the 3rd session, I'm going to show you that for nonlinear dimensionality reduction. And you can deal with missing data. So missing data, these all hold for factor analysis as well. I wanted to emphasize that. I look at PCA. I was at Aston when these guys did this work. And Chris Bishop was my PhD supervisor, so I tend to focus on probabilistic PCA, but there's a whole load of parallel work in factor analysis, which I want to emphasize exists as well. So here's that oil data. In this case, we've removed 10% of the values by missing at random, and you can do various things. You can do a little EM algorithm. There's a few other things you can do to fill in these missing at random guys, and you get a corruption of the original plot, but you can still see the structure. That's 10% missing, 20% missing, 30% missing. You can still see the structure. That's quite a lot of missing data. In fact, probabilistic matrix factorization can just be seen as Bayesian PCA. It's exactly the same model structurally, and that's being applied to deal with large data sets of preference data, collaborative filtering, where it's almost all missing data. Yeah, you've only got 5 values per person, and that's, in effect, this model with a very large amount of missing data. So probabilistic modeling gives you that ability to deal with that. Here's 50% missing. Still some of the structure there. Green's overlapping with red completely, but you can still see these separate islands. So factor analysis, as I've said, is the same model, but with this noise variance is now D. So each output now has its own variance, but it leads to this problem that you can't optimize this likelihood in one step. You can use an EM algorithm, but it's not the fastest way, and I did do a little bit of reading about it. It's just a massive field fitting these models. I mean, you'd really like a simple paper that just says what the best way is. I'd recommend, the thing I read that perhaps made most sense, R is the main statistical package. So a good way of understanding what the statisticians are doing is finding what command they put in R. And there's a way of doing factor analysis in R which describes how it's doing it, which doesn't fit easily how you're doing the maximum likelihood, so I can't exactly tell you how they're doing that. But I would have a look at that if you're interested in good factor analysis fits. I tend not to do factor analysis. I tend to do PCA, as I've said. Independent component analysis. So it's worth briefly mentioning that. So again, the same linear Gaussian relationship, but now these source distributions are non-Gaussian. And this has the effect of removing, well, it has the effect of messing up your nice analytical model, so you can't anymore write down easily the marginal likelihood and just do the fits. That's effect number 1. Effect number 2 is, instead of having a source distribution like this is like the example where, remember earlier I said with super Gaussian, if I said this is a 1,000-dimensional Gaussian, all those 999 dimensions are being projected on top of the origin here. Well, this is the equivalent plot for a super Gaussian source. Remember I said the same thing can occur. Central limit theorem shows you the distances are the same, but the points will lie along the axes, yeah? So this is the classical ICA type thing. If we were trying to do separation of sound sources, they're super Gaussian in nature. If you were to plot them, you would see these sort of crosses like this. The source distribution they use then has this cross form. Now, if I rotate it, when I rotated a sphere, it didn't change the sphere. So the density here, when rotated, doesn't change. But this density does. A cool thing to do if you want to do ICA, and you've got some standard ICA dataset which you know has independent components, just do PCA on it, project into the latent space, you will see a rotated cross. And ICA is just finding that rotation to unrotate it. Or you will see a rotated square. Sub-Gaussian sources do the same thing. So what they do there is in this latent space, the probabilistic interpretation, they do other ways of fitting ICA, but the probabilistic interpretation is you're fitting density which is non-Gaussian in this latent space to get your independent components out. Okay, so that's going to be it for the first session. So unlike Bernard, I'm not going to make you vote on, make the lazy ones say, yes, I want a break. You're all going to have a break, or you can ask me questions and I'll drink some water or something. Okay, so we'll restart in 5 minutes.